All right, guys, welcome back to part 2 of chapter 5. We're going to start this chapter by looking at the membrane function. So we know that membranes, plasma membranes, we went over those in chapter 4, they have a really big important role, right? They are basically the line that we draw when we draw a cell, just that circle, it's that line, that's the membrane, and it basically is enclosing all of the material of the cell within that membrane. And so the cell is kind of separated from the rest of the environment. However, even though it's separated from the environment that the cell is in, it still interacts with the environment. So there's still things that flow in and out. So materials and molecules and substances that go in the cell, and materials and molecules and substances that leave the cell. So the plasma membrane is a membrane forming the outer boundary of many cells. Okay. It consists of a double layer of fat. We know what fats they consist of. Remember, it was that phospholipid, those phospholipid fats that we looked at. So we have one layer of phospholipid fats on top and the other layer of phospholipid fats at the bottom. Each phospholipid fat has a phosphate group, which is hydrophilic, and a hydrophobic region, which are the actual fatty acids. So we have this double layer or a bilayer of the phospholipids with occasionally embedded proteins. So we have these kind of proteins basically embedded everywhere else, kind of just like a mosaic model, right? It's just like a mosaic painting or artwork. It just has random proteins littered throughout the actual plasma membrane. And these proteins are really important because sometimes we use these proteins to transport materials in and out. So the plasma membrane, it is selectively permeable, meaning that certain things can go in it and other things cannot, right? So it selects what can go through and what cannot. So if we want to directly cross, so just this blue part right here, if we want to directly cross the plasma membrane, only certain substances can do so, because this is what it means to be selectively permeable. The plasma membrane, this blue part right here, right, the actual phospholipids, only allow for certain things to go through. Those certain things are going to be lipid soluble molecules, or essentially hydrophobic molecules. Because if you remember, in this bilayer, we have this hydrophobic region. This is all fatty acids, right? This is a hydrophobic part. So in order to cross that, the lipid molecules, only hydrophobic molecules can pass it. And like they're the only ones that can pass it. Non-lipid soluble molecules, so basically hydrophilic or water-loving molecules, even though they cannot pass it. Sometimes they can if they're really, really small and they can basically finagle their way through the plasma membrane, but most of the time they can't. So a lot of times things like oxygen go through the membrane, bigger hydrophobic molecules can go through the membrane, but then we have specific things like ions. We know what ions are. Those are charged atoms. Ions, they cannot go through the membrane, because they're hydrophilic. Any other kind of hydrophilic substance or even large substances, they cannot go directly across the membrane. Okay, so the semi-permeable membrane allows certain solutes in and others aren't allowed in. It's permeable to hydrophobic molecules like lipid substances, but it's not permeable to larger substances that carry a charge and therefore are hydrophilic. Okay, so let's look at the actual type of transportation that we do have. The first type of transportation, and you have to also remember that molecules are always going to be moving. And so this is why we have transport in the first place. The first type of movement is known as diffusion. Diffusion is basically the movement of molecules to spread into available spaces. And they do so by going from an area of high concentration to an area of low concentration. This isn't too bad, right? We do this all the time. If I spray a little bit of perfume in front of me right now, if I just spray a little bit of perfume in front of me, that part where I just sprayed has a high concentration of perfume molecules. Eventually, if I just leave it, right, eventually these perfume molecules are going to be moving around and they're going to move to low concentration, so the rest of the room, basically, where there's low concentration of those perfume molecules, and they're going to diffuse and disperse in that way, right? So same thing, if I spray Febreze in that corner of the room right there, I will eventually smell it on this side of the room, even though it's across the room. I will eventually smell the Febreze molecules. That's because the Febreze molecules go from a high concentration over there to a low concentration of Febreze molecules over here, because I didn't spray any Febreze molecules. Okay, so diffusion is the net movement of like molecules or ions, solutes, not water, from a high concentration to a low concentration. And when we say if they follow this high to low concentration, you are following the concentration gradient, okay? And when you follow the concentration gradient, it doesn't require energy. So one way that I like to think about it is using a, I don't have a pen. Oh my goodness, I'm sorry. There we go. I found a pen. Is using the scenario of a slide, right? Everyone has used a slide when you were little. I'm sure you used a slide when you were little. I am going to draw it like so. That is my fancy slide, okay? We know that when we're little, we're always like lining up. There's a bunch of kids, right, at the top. There's a bunch of kids at the top. There's no one at the bottom because everyone wants to go back up again and go down the slide. And so this is you. This is you right there. You are going to want to go down the slide. You're going to want to go from an area of high people concentration, in this case, our molecule, right? And I don't know if you can see that, but molecule, put that in quotations. Our molecules are people. Here we have a high concentration of people. How about down here? We have no people. That means we have a low concentration of people or all of our molecules. So you go, when you go down the slide, that's you going down the slide, and you come here now, okay? So this person gets, you are now here, okay? You can go down the slide, and when you go down the slide, you don't need to push yourself down the slide. You just roll with it, right? Gravity does that whole thing with you. So when you go from high to low, no energy is needed. When you go down the concentration gradient, that's the same as going down the slide, and no energy, E stands for energy, absolutely no energy is needed. So this is diffusion. Okay, this is diffusion, and it happens naturally, and it's going from high to low concentration. Sorry, I should have shared my screen. There we go. Okay. So passive transport is a type of diffusion that allows, that basically allows a substance to cross the plasma membrane. Okay, so when diffusion happens across a plasma membrane, it's known as as passive transport. Passive because there's no energy, because you're still using the concentration gradient. An example is oxygen from our red blood cells to the rest of our body. The way we move oxygen through our body is thanks to passive transport. Basically what happens is that we breathe in a lot of oxygen, like the blood in near our lungs, they gain the oxygen in the red blood cells, and then when they move to our feet or to our hands that do not have any more oxygen, they realize that there's a high concentration of oxygen in the red blood cell, but there's a low concentration of oxygen in the environment of the hand. So where does oxygen go? It goes from high to low. So it goes out of the red blood cell, through the plasma membrane of the red blood cell, into the environment, because it goes from a high concentration of oxygen within the red blood cell to the low concentration of oxygen outside of the red blood cell. And this all happens until we get to equilibrium. So as you can see in this picture, we go from high concentration and it starts moving these this is just a dye. These dye molecules start moving around until they go to all the low places, like the low concentration places, and until they get to equilibrium. So everything is basically in equilibrium and evenly distributed. There's a net movement. The net movement basically ceases. Okay, so here we see molecules across a plasma membrane. We have passive transport or diffusion. This is high concentration. They're going to go to the right. The majority of them are going to go to the right until we get to equilibrium. And every time one red one pops over, this other red one from the other side is going to pop over the other way. And remember, this is like molecules. So in this case, we have many blue molecules on the left and like many red molecules on the right, but no blue molecules on the right and no red molecules on the left. So the blue are going to go to the right and the red are going to go to the left until both of them get into equilibrium. The fact that the other substance is doing the opposite thing is not going to affect them at all, because it's always between like molecules. There's a video for you guys to watch on the PowerPoint. I'm not going to play it here, but I highly suggest you guys go ahead and watch that. Very important video to watch. Okay, so as we were saying, not many things can pass the plasma membrane because the plasma membrane is semi-permeable, right? But what if we want to, the cell needs a hydrophilic molecule, or like a big molecule of glucose, of sugar. Sugar cannot pass across this plasma membrane by itself. It just can't. So in that case, it's going to need the help of proteins, of protein channels or protein tunnels. Basically, these proteins, as you can see in this picture right here, they form this channel. They're embedded within the plasma membrane, and they form a channel through the plasma membrane so that they have these hydrophilic molecules that would not normally be able to get through the plasma membrane and into the cell or out of the cell, depending on where they have to go. These molecules now have a nice little tunnel, a nice little cozy hydrophilic tunnel to go through in order to go into or out of the cell, depending again on where the concentration gradient lies. So this type of diffusion is known as facilitated diffusion because it's facilitated, it's helped along by the presence of proteins. Okay, so it's kind of assisted transport. So here, transport proteins, those proteins that form channels or tunnels across the plasma membrane, which are embedded in the plasma membrane, aid to bring things in. For example, water, right? Water obviously likes water. It is very hydrophilic. Water has a very hard time getting through the plasma membrane. And so it requires proteins to make it go inside the cell or outside the cell. We require water, okay? It's a very important thing. And so this water uses transport proteins to get in or out of the cell. Another example is glucose, right? Alone, it cannot pass through the plasma membrane. It's too bulky, it's too big. And so again, it uses transport protein. The facilitated diffusion has the word diffusion in it. So it's the same type of thing. It's still diffusion. It's still going from a high concentration to a low concentration, and it does not require energy. The only thing that's different now is that when molecules are moving around, they're going through the actual protein and not across the plasma membrane, because the molecule itself cannot. So in this example right here, as you see, I have 2 molecules of glucose at the top, 1 molecule of glucose at the bottom. So I have high concentration here, low concentration here. So where do my glucose molecules want to go? They want to go down, right? So in this case, I have a high concentration of glucose outside the cell and a low concentration of glucose inside the cell. So the glucose are going to go from high to low, from outside to inside in this particular scenario. So one glucose goes through, it goes through the through the protein embedded within the plasma membrane, because the protein itself basically makes a nice little hydrophilic tunnel for the glucose. It goes in, and then we arrive at equilibrium. Okay. So now moving on to osmosis. Osmosis is essentially the diffusion of water, okay? Because we know that in biology, cells, any living kind of organism is full of water, right? You are 70% water. Like there's a lot, other organisms are predominantly water. And so we have to think about the movement of water. And when we think about the movement of water, we can't think of the movement of pure water, because water in a biological organism is always going to have things dissolved in it. It's always going to have solutes in it. So when we look at water movement, we have to consider its solutes. So the actual water movement is known as osmosis. We don't call it water diffusion. We don't call it water movement. We call it osmosis. Okay? Osmosis is the movement of water. And I'm going to read the description and the definition of osmosis, and it's going to sound like it actually goes against diffusion, even though we know that osmosis is technically the diffusion of water. But it's going to for the first couple of seconds, it's going to sound very like contradictory to what happened with diffusion. So let's see. Osmosis is the net movement of water across a semi-permeable membrane from an area of lower solute concentration to an area of high solute concentration, right? And diffusion said it goes from high to low. And now osmosis is saying, wait, we're going from low to high? Correct. We're doing that, but it's actually the same thing as diffusion. So I want you to kind of look at it this way. I'm going to change. Okay. So let's pretend I have a beaker, a really big beaker. Okay, and in this beaker, it's separated by like a plastic kind of membrane. Okay, I'm going to see if I have. Yes, I do. I don't have the color blue here, but we'll do. Okay, so this green part right here is going to be my salt or my solute. Okay, so salt equals solute. It's a type of solute. So solute can also be a sugar or something like that. But salt is one of those easy things to remember that it means it's a solute. Okay, so this is as much salt as I have on this side. This is side A. Okay, and on side B, I have this much salt, just enough to cover the bottom. Here I have a lot of salt. Here I have low amount of salt or solute, correct? I pour in some water, and the water, even though I pour in the same amount of water, right? When I look at the water in relation to how much salt I have here, I technically have less water than I have here, correct? There's not that much water compared to side B. So I'm just going to write down here, I have low water concentration. I'm going to make these little brackets. These little brackets basically stand for concentration. I have low water concentration and on side A, and then on side B, I have high water concentration. Oops, you can't see that. There we go. So side A, low water concentration, side B, high water concentration. Now let's look at solute, because remember, we can't think of water in a biological system or in an organism without considering solute. In this case, our solute is salt, right? So here, side A does it have higher amount of solute or lower amount of solute than side B? Hopefully you say it has higher amount of solute, right? Honestly, it has like 1, 2, 3, I don't know, about 20 different salt molecules compared to 6 salt molecules right here. So this has high solute concentration. My brackets, remember, mean concentration. And here I have low solute concentration. Now, in terms of diffusion, okay, we know that things move from an area of high to low. So which way is in terms of, look at only the water molecules, because the only thing that's moving is water. Salts cannot actually move, okay? The only thing that's moving here is water. The solute never moves. So in which way is it going to go? It's going to go from high water to low water, right? Because it's just a diffusion of water. So it's technically going to go from B to A. If this was a porous membrane, if there was pores throughout this membrane, this water would go to the left until we find equilibrium, right? That's what diffusion says. It goes from high to low. Osmosis, again, is just a diffusion of water, but and in addition, it also describes it a little bit differently. Instead of saying high water to low water, it literally saying low solute, which means if we have low solute, it means we have high water, to high solute, which means we have low water. So that's what osmosis is. We go from an area of low solute to an area of high solute, and it's that movement of water. Both of them are going in the same direction. Okay, so osmosis is the net movement of water across a semi-permeable membrane from an area of lower salt concentration to an area of higher salt or solute concentration. Okay, you can also see it as basically the diffusion of water. Remember, a solute is just a substance that is dissolved in a liquid. So here you kind of see it a little bit more. So because of this, there's various different conditions that exist, right? We have various different kinds of tonicities of solution. We have a hypertonic solution, an isotonic solution, and a hypotonic solution, meaning a cell can be in a hypertonic solution, an isotonic solution, or a hypotonic solution. And what that means is that if a cell is in a hypertonic solution, okay, that means we're right here, scenario B. Oh, hold on, I forgot to change my camera. Okay, we're right here, scenario B, hypertonic solution. That means we have a higher amount of solute on the outside, okay, right? We have a higher amount of solute on the outside and a low amount of solute on the inside. See all those green things? We only have 3 green things in the inside of our cell, and we have a lot of green things on the outside of our cell. And we know that with water, it goes from an area of low solute to an area of high solute. Another way to remember this is that water follows salt. Water follows the highest salt, okay? So this means that in a hypertonic solution, the water inside our cell is going to go outside. It's going to move out of our cell. And because of this, the cell is going to shrivel up. So in a I'm gonna mime for you guys. I'm gonna be doing some terrible acting right here, and hopefully this will basically like burn this in your head so that you remember this for the exam. But in a hypertonic solution, the cell is hyper and shrivels up. Got it? You do it as well so that it stays permanently in your head. Okay, so in a hypertonic solution, the cell is hyper and it shrivels up. Okay, so what happens in a cell to a cell in a hypertonic solution? It shrivels up. This is why we cannot drink ocean water, guys. You ever heard of that? If you're ever stranded in the middle of the ocean, don't drink the water. Why can't we drink the water? Because the water of the ocean is way saltier than our cells, has a lot more salt than the inside of our cells. So when we drink the water and it goes inside the outside, like inside our bodies, which is technically the environment that our cells are found, all of our cells will lose the water because the water is going to It's going to follow that high salt concentration that we now have ingested in our body, and all of our cells are going to dehydrate. See? Applicable stuff that we find out thanks to biology. You're welcome. I'll just save your life. I'm just kidding. Okay, so in a hypertonic solution, that's basically us drinking ocean water. You dehydrate your cell. Your cell basically shrivels up and loses all of its water. Not a fun scenario. In a hypotonic solution, so see how we have an O now instead of an ER. In a hypotonic solution, now the things are a little bit different. We have a high solute concentration inside the cell and a low, only 2 versus 3, 2 like a small solute concentration outside of the cell. So remember, water follows the highest solute, so water is going to go inside the cell. That's a little bit of a problem, right? That's a little bit of a problem because when water goes inside the cell is kind of gonna swell up and blow up even. Okay, in fact, certain cells like animal cells, we don't have any extra protection outside the plasma membrane, right? We don't have a cell wall. We're not Groot. You guys remember that? We're not Groot. We don't have a cell wall, and so if water keeps going into our cells, the cells is going to expand more and more and more until it can't expand anymore, and then it's going to blow up. So in a hypotonic solution, our cells become hippos. Okay, got it? In a hypotonic solution, our cells become hippos. They swell up, and then eventually animal cells, so you and I, will blow up. That's terrifying. And then we have isotonic solution where we have an equal number of solutes outside as we do inside. And so the net movement of water in and out is nice and equilibrated and nothing bad happens. No shriveling, no blowing up, no nothing. This is good. You need to know hypertonic, isotonic, and hypotonic very well. Okay, so this is what's happening in an animal cell. We're going to look at animal cells, and then in the next slide, we're going to look at plant cells. In an animal cell, in a hypertonic situation, what happens? Hyper! So water goes outside and we shrivel up. Our red blood cells shrivel up. In an isotonic situation, look how beautiful and plump our red blood cells are. They're doing great. Whatever water goes in, a little bit of water goes out. They're doing fine. They're in equilibrium. In a hypotonic situation, it's terrifying, right? Because there's a lot of water that goes inside of our cells. The cells are going to really swell up and just become like hippos, hippo, and some of them are going to burst. We don't want any bursting to happen. So the control of water balance is a big deal, and this is known as osmoregulation. Now, in a plant, things are a little bit like easier for plants, right? If you think about it, plants really need water. You need to water your plants, something that I continuously forget to do, and that's why plants basically don't do very well in my house, because I forget to water them. Plants really like water, okay? And they can actually deal with a lot of water going inside of their cells. So in an isotonic solution, the plant is normal. In a hypotonic solution, when the cell blows up like a hippo, yes, the plant cell blows up and swells up, but because of that cell wall, right? The cell wall, which basically helps strengthen the cell. So plant cells have a plasma membrane, and then outside the plasma membrane, they have a cell wall. The cell wall stops the cell from blowing up, which is fantastic. So plants don't die when you have, well, I mean, you can overwater them, but that's just because you basically rot them. But you know what I mean? Like they're plant cells, they can basically take up as much water as they need. it doesn't matter. So they are very good in a hypotonic solution. And then in a hypertonic solution, the cell wall doesn't really help in this case, actually. The plant cell dehydrates and loses all of its water, and it just basically kind of, as you can see, detaches itself a little bit from the cell wall. This is actually known as plasmolysis. So in a hypertonic solution, plant cells are also screwed still. Okay, so this is what happens, different solute concentrations, different tonicities, hyper, hypo, isotonic solutions. Between an animal cell and a plant cell, you need to know both, okay? So animal cells, they blow up in hypotonic solutions. Plant cells, they just swell up and are happy. They're actually the happiest at that solution. And then lastly, we're gonna talk, not lastly, almost lastly, second to last, we're gonna talk about active transport. This is when we are actively pumping molecules across the membrane, okay? So back to that scenario that I drawn about the slide. Where is the slide? There's here. I have the slide. Sometimes we want to actually pump in something outside of the cell. So this is going to be inside cell, and this is going to be outside the cell. Okay. Let's pretend you want to go, and the cell really wants you back, right? You want to go back inside, but as you can see, so you want to go back to the top, which makes sense because you want to go back down again. You know what I mean? So as you can see, we still have a low concentration of the people molecule here compared to the high concentration of the people molecule here. So what's going to happen? If you were anything like me, I would literally climb up the slide, right? Climbing up the slide, like just holding onto the rails of the slide and climbing right back up. That obviously takes in a lot of, it requires a lot of energy. I don't know if I'd be able to do that right now, to tell you the truth. But I would climb up slides, and like that would require a lot of energy. And that's what happens with the cell. If they want to go from an area of low to high, because they really, really need that particular substance, they need to pump in that substance with the use of energy. So this is when we have active transport. You need energy because now you're going against that concentration gradient. You're going against gravity per se, right? As an example, you're going against gravity, which was basically against that concentration gradient, which was technically from high to low. You're supposed to go this way, but you're going the opposite way instead. And so in order, because you're going the opposite way, you're going through active transport, which requires energy. Okay. So active transport requires that a cell expends energy to move molecules across a membrane through transport proteins. So still here, we have the use of these particular proteins, right? These channels. So cellular energy, which is provided, we know what the cellular energy is like what it looks like, what the molecule is. It's provided by ATP. And this is used to drive a transport protein that pumps a solute or a substance against the concentration gradient, so against the gravity of that substance. Okay, the active transport allows cells to maintain internal concentrations of small solutes that differ from environmental concentration. You can compare it to rowing upstream. That's what your textbook says, and that's what I wrote in my slides. But I also like to compare it as like going back up the slide. Okay. So in order for active transport to happen, you need a transport protein, and you also need an ATP. You need ATP because this requires energy. Okay. So again, oh, I thought this was a video. It's not a video. Okay, so here you see you have low concentration versus high concentration of those orange substances, those orange solutes. Yet the cell wants this orange solute because it's greedy. In order for it to go back, to go from outside to inside the cell, this is basically going to have to go against the concentration gradient because it's going from low to high. So the cell gives ATP, gives energy, and the solute can go inside. So there's a few ways that we've seen transport now. We've seen passive transport. There's two types of passive transport. There's simple diffusion, which is just basically going across the plasma membrane, going from an area of high concentration of solutes to an area of low concentration of solutes. You also have facilitate diffusion, which is the same principles of diffusion, going from high to low. However, this time you are using an actual transport protein. So you're using a channel, and this is because the molecule that's in question that has to move in this time is a molecule that cannot cross the plasma membrane by itself, right? It's hydrophilic, or it's really bulky and big, and so it cannot cross like directly through the plasma membrane. And so this time it uses a protein transport channel. And then we also have the third option is active transport. This is basically going against the concentration gradient when we go from low to high, and we're still going through a protein. And since we're like a transport protein, and since we're going from low to high, it requires ATP, it requires energy. Now, last but not least, last but least in this case, we're going to talk about exocytosis and endocytosis, because sometimes our cells also like to shop in bulk. I don't know about you, but I like going to BJ's or I like going to Sam's Club, and I like buying things in bulk. It's a lot more convenient sometimes. The same thing with our own cells. Our cells are able to get rid of the stuff in bulk, and they are able to get things in bulk. This is known as exocytosis, exo exits, endocytosis, into, endo enters. Okay, so for larger molecules, transport works in and out of the cells. For larger molecules, transport works in and out of the cell differently. A lot of times these molecules are too big to pass through the membrane and too big to pass through transport proteins as well. Sometimes we want to take up more than one molecule at a time. So this is when exocytosis and endocytosis happen. So in exocytosis, this is the movement of materials out of the cytoplasm of a cell via a membranous vesicle or vacuole that fuses with the plasma membrane. So for example, this is the molecule that needs to be exported. You see how it's in a little vacuole, in a little vesicle. This vesicle is going to fuse with the plasma membrane, kind of bubble into it, and then puke everything out. So exocytosis is essentially puking of the cell. The cell is puking, okay? Lots of stuff are coming out, all right? So the cell basically regurgitates all of the inside stuff outside into its environment. Endocytosis is when the cell takes up a lot of stuff. And so it's also known as phagocytosis, which means cellular eating. This is how our immune system cells actually eat up the bad guys and the bad bacteria and everything, right? Because they can just eat them up. So here in this case, what they do is that these larger molecules that have no stinking way of getting across the plasma membrane, either directly or even with a protein channel, they're just way too big and they're too numerous. Our plasma membrane just kind of infolds and indents within itself. It kind of forms a little, starts forming a little vesicle, and anything that it gets, basically anything that is found within the little area that it's forming the vesicle gets plopped inside. So endocytosis is just sucking stuff from the outside, but big kind of molecules, okay? So like I said, an example is the immune system cells. It uses it to engulf and destroy bacteria and viruses. All right, so that is all that I want to go over with you in chapter 5. The last video is a fantastic video on membrane transport. Please watch it, and I shall see you guys in the next chapter, chapter 6. Bye, guys.